Welcome, Sham Stars. Under the heading of Chapter Cell, the Unit of Life, today we will discuss about cell envelope and its modifications, ribosomes and inclusion bodies. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. That is the outermost glycocalyx followed by the cell wall and then the plasma membrane as shown in the slide. Although each layer of the envelope performs the distinct function, they act together as a single protective unit. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is semi-permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, or lamellae. They help in cell wall formation, DNA replication, and distribution of daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts as shown in the slide as well: filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outside. Hook is the curved sheath which connects filament to cell. Basal body anchors the flagellum into the cell wall and membrane. Now let's talk about ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with plasma membrane. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the messenger RNA into proteins. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bounded by any membrane system and lie free in the cytoplasm. For example, phosphate granules, sulfur granules, cyanophycin granules, and glycogen granules. Gas vacuoles are found in blue-green algae and purple-green photosynthetic bacteria. So children, this is all about cell envelope and its modifications, ribosomes and inclusion bodies. Tomorrow we will be continuing with the eukaryotic cells. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.